All right, good afternoon, everybody. Hope you enjoyed lunch. We're going to be starting session 3 now. So first, I'd like to introduce our session lead, Dr. Ping Du. Ping has worked for 5 years in the pharmaceutical industry and worked with different dosage forms, including MDIs and DPIs and different administration routes. Right now, Ping is a reviewer in the Office of Bioequivalence, Division of Bioequivalence 2, Office of Generic Drugs at US FDA. So please welcome Dr. Ping Du.
Thanks for the introduction, Ross. Good afternoon, everyone. Welcome to session 3. Slightly different from the agenda, session 3 combines MDI and DPI into one single panel separated by 30 minutes break into 2 parts. The single panel will discuss based on topics. So feel free to ask questions during the panel discussions. We will keep monitoring the chats. The introduction here will be very brief due to time limit, and there are 11 panelists and 2 moderators to introduce. So in part 1, the moderator, Dr. Diana Vivian. Dr. Diana Vivian joined in the Division of Bioequivalence 2 as a primary assessor in 2014, and he has served as the Associate Director of DB2 since 2019. Dr. Vivian has BE interests in diverse areas, such as complex topical dosage forms, nasal and inhalation products, and the BCS system. Panelists. Dr. Bäckman is Senior Advisor at Bäckman Consulting AB. He provides advice on planning and execution of clinically relevant test programs, as well as the application of PBBM to understand the relationship between in vitro drug performance attributes and clinical performance. Dr. Mark Lepore, he began his career as a physician, board certified in both general pediatrics and allergy, clinical immunology. He is currently the CMO at TransBio. He has authored numerous papers in the respiratory space and has been a co-author for medical textbooks. Dr. Markham, he serves as FDA supervisory physician and director of the Division of Therapeutic Performance 1 in the Office of Research and Standards, Office of Generic Drugs at FDA. Dr. Markham has been at FDA since 1998, and he has served various roles in US FDA. Dr. Qing, she is the Deputy Division Director in the Division of Bioequivalence 1, Office of Generic Drugs at US FDA. She has over 10 years of experience in the assessment of BE for NDAs, controlled correspondence, citizen petitions, product-specific guidance, and protocols. Dr. Singh, he is a Senior VP and has headed the CIPLA Respiratory Center of Excellence since 2013. Earlier, he has spent many years of his professional life at the US FDA, where he was also the designated expert on respiratory drug products in the Office of Generic Drugs. Dr. Yuchun San, he is currently working at Apotex as CSO, Biopharmaceutics and Biostatistics. His main responsibilities are to provide PK and statistical advice in preparing protocol and study reports for PK, PD, and clinical studies of complex drugs and biosimilar products. Dr. Liang Zhao, he has been serving as the Division Director of Division of Quantitative Methods and Modeling, Office of Research and Standards, Office of Generic Drugs, US FDA since 2015. Through his 16 years professional career, Dr. Zhao has established his leadership in industrial R&D, quantitative methods and modeling, and model-based strategic decision-making in regulatory and industrial settings for generic and new drugs. We also have Dr. William Ganley and Dr. Xian-Ming in part 1 panel discussion, and they have been introduced in previous sessions. In part 2, the moderator is Dr. Darby Kozak. He is the Deputy Division Director for the Division of Therapeutic Performance in the FDA's Office of Generic Drugs. Dr. Kozak leads development of new analytical methods and equivalence evaluation methodologies for complex drug substance and parental, ophthalmic, otic, and inhalation formulations. The panelists, Dr. Abhishek Gupta, he is the CSO of Transpire Bio. Dr. Gupta is responsible for providing the strategic and operational leadership to develop and implement the company's scientific strategy, overall product portfolio, and introduction of new product opportunities. Dr. Banaszak Holl, he is a professor and associate dean for research in the School of Engineering. He has more than 20 years of experience in polymer and other nanomaterials for drug and gene delivery, including detailed characterization studies. We also have Dr. Bäckman, Dr. William Ganley, Dr. Markham, Dr. Singh, Dr. Diana Vivian, Dr. Xian-Ming Zeng, and Dr. Liang Zhao, who I introduced in part 1 panel session. They also will participate in part 2 panel discussion. And then we will have the summary of session 1 and session 2, followed by our panel discussions. Thank you very much.
Right, good afternoon, everyone. Just wait for a second before I switch to the other presentation. Yep, so this is, I'll give a brief summary of our discussions that we had during session 1, which was focused on the in vitro studies and the alternative approaches where these, again, fit in, and trying to tackle some of the larger questions as to, if you're dealing with suspension MDIs and DPIs. So in this first part of the session, this discussion was, or the presenters here were the ones that were involved for the dissolution and realistic APSD discussions, which gave us a good evaluation of a lot of the different considerations that go into the different study methods, into the statistics where different products might be able to utilize these approaches, and certainly gave us a good understanding of the complexity of when you're conducting these studies, how you might want to design them and what considerations you need to put into when you're developing these methods. So in our small group discussions here, the first bit of information, this is just kind of like a snapshot of some of the discussion we had for the dissolution. Hold on. Yeah, so this, so for the dissolution discussion, in general, across the tables that we had, we felt like this type of method was really independent of the dosage form, so it'd be applicable for both MDIs and DPIs, and kind of no surprise here, the API solubility could be helpful as a guide for trying to understand, again, if the dissolution method's going to be useful here. However, there was some points that it may also be useful for some high-soluble APIs in terms of being able to inform on their microstructure. So, it might not be so clear-cut to rule out high-solubility APIs with dissolution. Again, it relates back to the purpose of why you're trying to do the study here. Certainly, the formulation itself would be able to play a major role, and so it's, in addition to the API's characteristics, you can't rule out the effect of the formulation. And in terms of the, like whether or not there's a preferred dissolution apparatus, there was some input that the paddle over disc is one that's more conventional or has been used more often and using with sink conditions. And this can be applicable for API. So one of the potential methods, again, that might be useful. And then aside from bioequivalence, of course, there was some thinking that dissolution could help with as a quality tool for batch-to-batch variability. But more specifically for its role in alternative approaches, it could be used as a way to assess for potential bioequivalence failure mode for these types of products. In terms of its roles, it could be used for evaluating dissolution-limited APIs, assessing exposure. It'd be useful for input for modeling PK and post-deposition behavior, and then establishing IVIVCs that can inform on the formulation microstructure and impact of release. So there is still some points about that, in terms of sample collection, what might be the best way to do this to avoid the effective dose. And then, also some thoughts on if there's a way to develop some type of BCS system to be able to try to inform on, again, which types of products might be more, I guess, useful or would see the dissolution as a more useful test. So after this, we had another discussion on can you go to the next slide? Yeah. So that we had a discussion on realistic APSD, and just some general comments across that were certainly the challenges that can come from it is related to the study size. So again, number of mouth-throat models and inhalation profiles. If you're trying to do a wide-ranging study, this creates a lot of NGI studies that you'd have to do. So that's certainly something that's challenging. The models themselves, their variability can present challenges, and the way these were designed, it wasn't that these models were paired with the clinical study, and so like if you're trying to use these published clinical data, that data wasn't done at the same time or helped to develop these models. So there's some challenges with relating the results for the RPSD directly to the clinical data, and trying to understand again, whether or not the relationship you're seeing is going to be meaningful here. Because of the variability in these models, they might not be really suitable for like a QC tool. But on the other hand, they could be useful for device optimization. Also, as well as like if you were to do a type of propellant change, maybe they might be useful there. And then in terms of some formulations that have high FPF or higher efficiency formulations, these might be able to detect or be sensitive to these types of dosage forms. So as you increased your FPF, you might see more utility out of using these models. There was some thought on using or considering realistic APS as a primary study method, and in addition for areas of additional input, focusing more on wanting more specifics on how to do the actual method development and validation, selecting of parameters, some additional points here for number of shot weight and number or appropriate shot number. So today we looked at, or we had presentations that were focused on particle morphology, and as well as like other potentially in vitro study methods. So for there, when we went into our small groups discussions, in particular for the particle morphology, the thinking was that it could be more important for the fine particles, which might be more impactful for PK. Again, the applicability for particle morphology would be useful for both MDIs and DPIs, and in terms of its the results from this, it could be useful for evaluation of API and excipients. It could be useful for informing on aerodynamics and dissolution performance, but also looking at more understanding morphology in terms of its role in product development, in addition to performance testing. However, the consideration for what type of statistics to work or to use for this was a bit of a discussion. The thinking was maybe if statistics were going to be used, it'd be more appropriate for like for the performance versus like at a developmental stage, because you want to provide some flexibility during the product development. So it might not be as maybe like applicable at that stage. So again, trying to understand where best to incorporate statistics for this type of performance testing. In general, for the alternative approach, the thinking was maybe it could be more of a supportive role, but again, not necessarily useful on alone as a standalone study. It would be let's say dissolution and APSD might be a more critical test. So you'd want to utilize morphology to help explain or understand if you saw differences in dissolution, for instance, instead of just looking purely at morphology. For additional inputs, maybe some thoughts on what's the best way to prepare your samples here. What specific morphological features you might be interested in. Again, because there's lots of different ways you can characterize it, even depending on the type of test you're doing. And then, like I was mentioning earlier, the type of statistics that are being used or could be used here. For just in general, other types of in vitro tests, there's a lot of discussion on the evaporation rate and velocity profiles. So trying to understand how best these can fit. So thinking here, some thoughts were that maybe it might not be useful if your formulations are already Q1, Q2 the same, and you had a similar device. It might be a more useful test if you have a non-Q1, Q2 formulation. In general, some of the thoughts for the evaporation rate and velocity profile. Again, it might be more useful as a developmental tool and for validation of in silico methods, in trying to understand again the role of the device that's going to play into this. So it's when you're trying to understand like you can't just purely look at the formulation as this interacts with the device. There could be components of the device that can influence your evaporation rate and velocity profile. So essentially, you wouldn't just be able to rely on your formulation sameness as a way to support equivalence here. You would need to have equivalence between a test and a reference. You might need to have a characterization of the characterization of the difference evaporation rates between the test and reference. Let's see. So for pre-actuation characterization, the thinking was maybe this might be more valuable for DPIs instead of MDIs. There could be some, so DPIs have bulk, so in terms of like the packaging of the formulation, there might be some additional considerations that go into those types of, I guess, manufacturing processes. So because those might be influential in their performance, then this type of pre-actuation characterization could be informative. Whereas for the MDIs, the thinking was that maybe pre-actuation characterization might be able to be covered through quality-related tests, somewhat related to like stability, and there's also just the general feasibility concerns of being able to conduct this study. So there could be, again, there might be other post-actuation studies that can inform on this aside from a more specific post or pre-actuation characterization for the API suspension. So it may not be like in this case needed. So lastly, there were some thoughts on if you, and so this is more like a general comment, but if you have tests that, if across the different in vitro tests, if you have some that don't necessarily pass, being able to utilize some in silico models to help support your overall BE decision making. So it's trying to again utilize in silico methods to help support your overall bioequivalence. Can you pull up the slides again, just a sec? Yeah. So in terms of the role for autonomous approach for the evaporation rate and velocity profile, this was viewed as more supportive. And like I said earlier, the pre-actuation characterization, again, maybe more useful for the DPI formulations, namely, again, because of the impacts from their type of bulk packaging and such that could be impactful in their performance. And then likewise, some discussion on need for like what type of statistical methods you'd want to use for these, because particularly for the evaporation rate and velocity profiles, those can raise a lot of questions as to the best way to do those studies, and then because of which, again, what would be the best statistics to use for that? All right, thank you very much.
Okay. All right, like Bryan did before, I'm going to be providing an overview of session 2. So I really enjoyed the session overall. We had a few talks at the end of yesterday. The first 2 talks were from Dr. Omar Osmani and Dr. Heino Stas about using charcoal block PK studies, and both of them also included in vivo imaging, which I thought was interesting. Myself and Dr. William Ganley spoke about model purpose and model selection. And for the discussion, I thought we had a really, so for anyone online, I thought we had a really lively discussion for just all around. I was really kind of happy to see how people were involved. So for alternative PK studies, I'm just going to try to cover all the different viewpoints because there wasn't always agreement on these viewpoints. There was general support for using charcoal block PK studies, especially if you've got a compound with high oral bioavailability, where that charcoal block could really distinguish between lung and gut absorption. There was expressed a need for charcoal block validation to make sure that the block is actually doing what it's supposed to do. There were some concerns about the ability of subjects to ingest a consistent dose, because Dr. Usmani did mention that in some cases, even though they were trying to administer, say, 200 to 300 millimeters, some subjects could only ingest like 10. So apparently there can be some issue with that. And also there is sort of a lack of an established dose, like how much of this should you give to everybody? As for using an IV arm as part of a PK study, nobody thought that this should be part of a BE study. Some people did argue that it could be useful for providing clearance data for modeling. Others said that the inhalation PK data would be sufficient on its own. We talked about the use of gamma scintigraphy with the charcoal block PK study and noted that even though this may not be useful for establishing BE due to legal ramifications of tampering with the product, some thought it could be useful for supporting modeling efforts and trying to understand how PK and regional lung deposition relate to each other. We also talked about the use of alternative PK studies besides charcoal block. And so there were some suggestions about partial AUC metrics being used from a PK study. Someone mentioned something about using a methylcholine challenge along with the PK study, also using a PK study to investigate the impact of different flow rates. So the next small group discussion was on model selection and model purpose. So there was a lot of discussion on this first point, as you can see. This was on the role of modeling for supporting generic orally inhaled drug products. I think everyone agreed that they're useful for product development, but there was kind of a spectrum of opinions about how useful this is for an alternative BE approach. Some thought that if you can pass the wide variety of in vitro studies that are being proposed, that should be enough. Others thought that you could use the model to establish a biopredictive in vitro and or in vivo study. And then at that point, once you've established that as biopredictive, you don't necessarily need to keep using the model. And then others thought it could be used directly. So that was the kind of range of opinions. Everyone basically did agree that advances are needed for model validation. A little bit further on this topic, for models that were used to directly support BE, we talked a little bit about intersubject variability, intra-subject variability, and that this can be accomplished for the deposition models by having a variety of patient-specific lungs. Talked a little bit about the difference between semi-empirical and CFD models, and the runtime factor is a big consideration here, just because CFD models can take a very long time. And semi-empirical models can be used to, you can do like, say, 1,000 semi-empirical simulations in a reasonable amount of time. But there's some advantages and disadvantages to both, and it was proposed that maybe using these together could be useful, particularly for the semi-empirical for the smaller airways. And then thinking about PBPK, these were agreed to be useful for low solubility compounds, especially where that connect between regional deposition and regional absorption may not be that clear. or also for a limited transporter-mediated receptor binding, kind of the same situation there. Okay, and then coming into this morning, we had a few talks from Dr. Jan de Bakker and Dr. Jürgen Belita, Dr. Kevin Feng and Dr. Qing Lin. So the first two were from Kevin Feng and Jürgen Belita on the use of modeling to establish a link between PK metrics and regional deposition. And then the following two talks were on validation of regional deposition models and also just some of the other ways that these models can be used. So the discussion on PK metrics to inform on regional deposition. So thinking about establishing this link, there was a kind of general opinion that using these population PK and PBPK could be useful. Some thought it could be used in a complementary fashion. Others weren't necessarily so sure about that. The choice between modeling types could be dependent on what you've got for your available in vitro data. So depending what data you've got, one might make sense over another. But we thought that for the future, these methods would potentially establish a non-compartmental approaches like, say, partial AUC or Cmax or something like that. So if you could do it successfully, the goal would be to have some kind of non-compartmental metrics that you're using to establish B, and the model was just something that you use to kind of help get you to that place. There was a consensus for model validation, obviously important, but some people offered some caveats that the lung PK data are not typically available, so that would be the ideal data source for comparison, and also animal studies may not be too helpful. As far as what types of compounds might be useful for establishing a link between PK metrics and regional deposition, pretty much everybody thought low solubility drugs would be useful here. Also thought that slow dissolving drugs with rapid permeability may be the best candidate, and some thought that rapid dissolving drugs with slow permeability might also work. As for model validation, we talked a little bit about do we need to validate with a non-BE orally inhaled drug product to establish model sensitivity? And some thought maybe not always necessary, and others thought that if you establish an IV-IVC, it really could help improve your chance of success. And as far as statistical comparisons, we didn't talk as much about this, but there was an opinion that we could use the kind of general 80 to 125 limit for the regional deposition, and then whatever link you've got, so you would kind of carry that through to your PK metric so that the PK metric limit would reflect that 80 to 125 limit in regional deposition. And then the final discussion was on regional deposition prediction validation. And we talked a lot about the division between central and peripheral regions because there really is a lot of kind of disagreement in literature about how you do this. We noted that there is this disagreement, but one approach is just to make a decision and just be consistent and just use it in all cases. And that could be really helpful actually if it came from FDA that we were saying that this is the way to do it. It was also mentioned that even though in theory the division between central and peripheral regions is important, it might not always be the case. It just hasn't necessarily been proved as such, but if it can be disproved then for a certain product, then that might not be important, just something to think about. And for CT scan resolution, this can be an issue for CFD studies because sometimes they don't cover as many areas as you need, so you might need a complementary model. And another person mentioned that actually central and peripheral may not be enough. You may need to divide between proximal, central, and peripheral. And we talked about mapping between three-dimensional lung deposition predictions to 2D space for comparison with in vivo imaging data, and pretty much everybody thought this was a good idea. As for acceptance criteria, we thought when you're comparing their deposition predictions with in vivo data, this can be complicated if the in vivo data are especially variable, and it really might depend on what your in vivo data look like. As for validation, some people thought this would be best with 3 different lung models. Others said that you should make sure that you're validating against mouth, throat, and lung deposition. And finally, we talked very little bit about benchmark validation and credibility standards, and we kind of all agreed that that was valuable. And so that pretty much summarizes session 2. Looking forward to the panel.
So now it's time for our part 1 panel discussion. And our panelists, please join in.
Test the weight limits of the platform. That's good.
We have one more seat down here if you guys need another seat.
You're the moderator.
Okay, thank you to our panelists for coming up here. We're going to start with one general question to get discussion started, and then if anyone in the room has questions, please feel free to use the microphone and come up, and we will also be taking virtual questions. So for our first question, what are the current challenges or gaps in research with alternative approaches to the CCEP study for MDI products? Would anyone like to start with that one?
Can you also put the question on the screen, or there's no way to do?
I cannot. I'm sorry. And you don't have to encompass all of the challenges, just what you think may be some of the major ones.
First of all, I would like to commend and congratulate the FDA and CGRS for a very comprehensive, long-awaited conference. Because this kind of conference is very rare, where there's an open discussion. It supersedes all the in-person meetings or product development meetings, and I think, and the variety of experts that I saw is amazing. So once again, thank you very much, FDA and the CGRS, for the opportunity. Now,
Well, I wanted to point out that it's the participants that make the meeting. So thank you to all the participants who took time to come to this meeting in person, and for all the folks that are joining us online. Thank you.
thank you. And I took the courage of taking the mic in my hand because I have worked on both MDIs and DPIs. Some of the challenges are common, and the most significant, the number one challenge is we are waiting for precise guidance from the agency. Now, there's only so much one can think of, and people have done their best effort. In my sort of last 5 years, I have tried to cover almost everything that has been discussed over the last 2 days. And there are certain experts here who have worked with us. So therefore, I think number one challenge is knowing exactly what we want to do. And a lot has come clear in this workshop, as some will be summarized later on. Second is, if we look at with this, that is the advice related to both what exactly needed to be done and what kind of statistical method analysis needs to be done. Because if we define the method, don't tell what the limits are, the stat is, then industry is at a loss, and many times the attempts can be failure. Since I have been on the both sides of the fence, I can see issues that can go on both sides, but that's where the direction is, and I'm glad in this workshop it's happening. And the second is that we are dealing with a new technology in a number of areas. And the validation of method on new technology is a challenge, as it has been discussed a number of times. But if I look at the generic industry development from after Hatch-Waxman was passed, that act was passed, what were the hurdles in development, how it came? Initially, there were 1 or 2 CROs, then there were many CROs, then companies started doing their own studies. I would like to request experts to adopt a pathway where the conduct of the study is not limited to 1 or 2 CROs. So whatever we choose, it's not that certain CROs who gain one or the other way certain knowledge, and that's the only. Let's see what is must to have, not nice to have, and bring all the sophistication in it. So I think these are the challenges main are, the industry wants to understand. Third is that the time is going to take to bring the information and the cost versus doing the clinical study. So a clinical study, let's say on a given product, let's say if it's a $20 million, and if now we include each and everything that we talked about, the cost and time of doing these alternative BE approach might exceed the clinical study. And the risk of failure is much more here than the clinical study. In the generic drug clinical studies on inhalation product, a study clinical should not fail if the product is good because of the, talked about lack of dose response. So all these considerations, how we can, expedite our decisions and pass it on to the industry. Go ahead, please.
Thank you.
We should bring the question back to the MDI. It was like, what is missing in the MDI space, right? We currently, I think at this meeting, we split the MDIs into suspension MDIs and solution MDIs, and each of those have perhaps a different approach. And then we also heard that there are some, when you actually do realistic APSD modeling, there are some gaps there in the knowledge as well. So even though we're recommending those in our product-specific guidance, so I'm looking at you maybe for some thoughts along those lines.
Yeah, so thank you for the question. So the question is about the challenging of developing MDI products. I thought of my time over 5 or 6 years ago, and we developed 2 MDI products, solution products for UK. One actually is right now on the UK market, serving the patients. We also successfully developed a suspension MDI product, which was much more difficult because of the issues around the PD study. And that product right now also is serving the US patients. In the meantime, I remember we developed a suspension formulation that is a combination product, and based on the alternative approach. We passed the PK, we did the CFD modeling, we did the MDRS, we did the dissolution, and we also did the PBPK modeling, pop PK modeling, and the PBPK modeling. Every single box has been checked. I believe that product is still being under review. So I think the biggest challenge for the industry is really what I've been talking about over the past 2 days, the clinical endpoint study. That is our biggest challenge. I think we are right now, we are working in the right direction. I think I want to echo what Dr. Singh just expressed. I think this is the first time I believe that, I have been interacting with the FDA for many years. I think this time I feel that the FDA is really willing to engage and listen to what the industry is talking about. I think this is a great sign. I really appreciate your efforts, the FDA. Congratulations, thank you. Hopefully we will be, FDA will be implementing a more scientifically rigorous and a pragmatic approach and so that we can develop more generic inhalation products to serve the patients of the United States. Thank you.
Thank you for those perspectives. Does anyone have any thoughts on what are our current research gaps that would help us close that gap toward a more clear framework for approval of MDI alternative approach?
For me, I've listened to many interesting talks through the conference and perhaps different way of developing the so-called model to predict lung deposition. My feeling is that perhaps we are trying to look for that perfect model to look for or to check for microscopic magnitude of difference. And yet, the therapeutic window of the drug that we are looking into have very wide margin, safety margin. So I'm always have a question as to, are we putting our effort appropriately? You know, knowing that patient needs affordable generic quickly. We cannot wait like 5-10 years of research to develop that perfect model, you know, that allow you to look for small differences, and yet the therapeutic window is so wide for the drugs. So I mean, that's my perspective from a generic company.
Thank you.
Can I add to that, to your question also? Yes. In the context of MDIs, we have a Q1, Q2, a lot of projects for guys is Q1, Q2. Does anyone here want to comment on non-Q1, Q2, maybe even in the context of new propellants for MDIs, things like that? What knowledge gaps do we have there? We've discussed some of that in the course of this meeting.
I think one of the things mentioned was the cost and the time, once again. And yesterday I was just contemplating from what we have been doing is just to support the alternative BE approach for his product that has two strengths, higher and lower. Just the realistic in vitro testing, cascading practice, it accounts to about 2,000 NGIs. And that goes to about $3 million by itself if you get it done from a CRO and all that. But nonetheless, coming back to, I would hope that the agency sticks to the Q1, Q2 sameness. That alleviates number of other risks or some additional work somebody will have to do. Regarding the propellant change, I think the history of FDA is that it's a 505(b)(2) approach, pathway, unless now OGD takes the responsibility, because this is a 99% and above excipient, which is going to change. It's a functional excipient. And with the change also, there's coming a change in the valve, gasket. I mean, the container closure system is going to change. It's not just the propellant which is changing. So therefore, I personally, can't think that this will come into as a generic, but all the knowledge and the base that we are developing, OGDs are developing, it might help when it gets translated by those 505(b)(2) applicants to the new drug. They can say some of the things can be demonstrated this way, and there may be less clinical burden over there. That's the only thing I can imagine, unless OGD today decide that this is a generic product with the change of the propellant. Thank you.
We have a counter to that?
No, actually, you ask about the new propellant. That's where I think the stepwise approach doesn't apply. Because if you have a new propellant, the evaporation rate will be different. So if you have a different evaporation rate, even particularly for solution formulation, for example, then the droplets will have different physical chemical characteristics. For example, if the new propellant evaporates faster, then the droplet, the particles could be more amorphous. then you could end up with a slightly different dissolution rate. Now, if you have a different dissolution rate, so according to the European approach, stepwise approach, if the PKBE, even if you can demonstrate the same PK profile with a new propellant and the current propellant, then since the particles' physical properties are different, the dissolution rate different, that means the deposition profile will be different. So that's where with the new propellant, you really can't. I don't feel that the stepwise approach is sufficient. And even if you have demonstrated this propellant is safe. Now, in terms of the same propellant, Liang Q1, Q2 formulation, Markham, you asked, I feel that depending on how different your formulation is, if you Liang Q1, Q2 formulation, if the difference is not significant, then you can adjust the container closure system, adjust the actual orifice size, etc., to make the droplets or spray pattern prone geometry similar. I think then you still have a chance to demonstrate the bioequivalence with the current with the approach we are talking about.
Can we hear from some other folks?
Yeah, perhaps I can talk to that. So within the context of an alternative approach where we're talking about all these advanced in vitro tests and in combination with modeling as well, I think for a non-Q1, Q2 formulations, we've got an opportunity to identify what the key performance attributes are and what actually influences, exposure at the site of action. And I think if we can use all of the technologies that everyone here are experts in, that we've been discussing over the last few days to do that, then there's a potential that we could maybe go through a similar approach for non-Q1, Q2, or maybe with an abbreviated clinical endpoint study or something like that. So I think that is certainly open for discussion. Yeah.
Thank you. Are there any questions from the audience? We have a microphone here in the back if anyone would like to ask a question to the panel.
Thank you. Just on that point, though, I think the Q1, Q2, and maybe this is the time to bring in the device. So Q1, Q2 deals with the formulation, but it's a combination product. So there has to be some element of assessing the impact of the device on the product performance as well, and where that comes in, and maybe a few comments around where that sits relative to maybe more downstream testing.
And just to note, the MDI devices, there are not a lot of variation to them, right? But there are some new technologies coming down the line for some inhalers.
Does anyone have thoughts on how device sameness should be evaluated?
Not really device sameness, but if you think about device performance in the sense that I think most of potential differences that are clinically relevant in device performance is probably captured by the extent that it varies the lung dose and the aerosol particle size distribution of the lung dose if you don't have a dissolution rate limited. So just looking at the device part, I suppose you can imagine that different devices and different propellants will have differences in lung dose. But I think that there are potential, or there are in vitro methods that could assay that, as well as deposition modeling efforts. So I think one could go down that road potentially.
Just for MDI, we know for MDI device, it's a canister valve actuator. For canister, as you know, the surface coating is critical. For the valve, the elastomer is critical. For the actuator, we took sunk volume, land length, and the orifice size are all critical. And if we control the critical parameters for each device component to be similar to the reference products, in conjunction with the Q1, Q2 formulation, and also Q3, if we can identify Q3 parameters, I think the combination of Q1, Q2, Q3, similarity in the device and container closure system, we have the same product.
Okay, one more audience question I see.
Since the methacholine challenge method for albuterol is proven to be very efficient and cost-effective, I'm wondering why the guidance for ipratropium and tiotropium muscarinic antagonists don't use that same system when those drugs are specific antagonists of methacholine?
Yeah, we evaluated those data internally. Actually, we have in the day one, there is a presentation about the flat dose response curves. So whenever there's a feasibility, we would do the dose scale conversion. So it depends on the characteristic of the dose response curves. For albuterol, it's a case, and that fit into the feasibility in terms of practice. If the dose response curve is too flat, basically a little bit of deviation in the PD response will lead to high variability in the converted dose scale. So that kind of will give more uncertainty rather than solving the problem.
Yeah, I just want to point out that the product-specific guidances might say one thing, but if any sponsor or company has a different approach, they're free to come in for a meeting with us, a pre-enda meeting to discuss an alternative approach, present some data saying, hey, this might be a viable pathway forward, and we would consider that in the context of the development program, in the pre-development meeting. Thank you.
Confirm what Markham said. You're welcome to present data that would contradict, could make the B assessment even more efficient.
I've got a non-industry response for you, Les. So I think maybe the question's a bit more fundamental. It's really, should we be doing PD studies at all, methacholine studies at all? Are they solving the problem? And I think that's probably in large part what this whole conference was about, right? To me, anything that's FEV1 based, which includes methacholine, is not giving us the answers we seek. It's not filling the gaps. It's not, as Dr. Newman mentioned, it's not addressing that residual uncertainty. So if we can't get information on what's happening locally, regionally, within the lung, I think we really have to be thinking about whether these approaches are viable at all. Other than for research purposes.
Can I make a comment to that? The selection of the methacholine challenge design for albuterol by agency was done after looking at a number of, actually several designs, which was conducted at Johns Hopkins. But nonetheless, I think we have to look at something that, how do we diagnose certain diseases, asthma, COPD? The fundamental parameter is FEV1. I mean, I know their variability, but methacholine challenge and the rescue medication has two indications. One of them is prevention of the bronchospasm, and the second is that if you are exposed to certain adverse environment, then albuterol will relieve it, or if some of the tiotropium will do that, or the SABA will do it. Then the other, why is this conference here? Why is OGD considering making all this investment time and having the money that given to research grants is that they are kind enough to consider of waiving the clinical study because it doesn't tell you anything about, sensitivity of the method. And among various, the methacholine challenge was found to be sensitive, that you get different response with different doses at single and multiple doses. And the cost of conducting a clinical study based on methacholine, even in today's day, it would be still much less than what we will spend on various aspects, which we are not sure on the non-clinical approach. Number one. Second, all albuterols are genericized. So that is gone. Now, if you want to apply that to SABA or SAMA or LAMA, it's a very interesting question because it directly relates to the mechanism of action, which is cholinergic. And if I were to make a decision for a sponsor, if I have to advise, I will look at the cost and the time it's going to take. And in my opinion, for where the challenge design can be used, it is for single drug, then it's a very efficient design. So it is directly related to the mechanism of the action or what the clinical effect of the drug is. So therefore, slight difference of opinion here. So, okay.
Thank you.
You know, we have tried that methacholine bronchoprotection model many years ago, probably over 20 years ago, on corticosteroid. We just found it data being so variable and so not predictive, and we just kind of gave up on it. I have to say.
Inhaled corticosteroids, but there is not a flat dose response with ipratropium and
Yeah, I think we also try it on salbutamol, right, which is same as albuterol in US, and the data just seem to be too variable.
I disagree with you. In fact, the 3 albuterol generics that have been approved by this agency were based on methacholine challenges that showed a clear dose response. So it may be how you're doing it. It may be who's mixing up your methacholine solutions. If each study site has to mix up their own solutions, you get a tremendous amount of variability. There's a lot of issues that have to be attended to get a good quality study.
Yeah, I think we internally looked into the data. Actually, before you posed the question, others already collected some data. I kind of agree with Dr. Tang that the data is very variable. If you, for certain drugs, in theory, it could be you're expecting a dose response curve in terms of a methylcholine challenge. Then many times in practice, you do observe a high variability. Like if you convert the PD20, put that as the response against the dose, it's very flat, very variable. Then basically you cannot characterize the curve. And also it's flat, little bit of change in the PD response will convert to a large dose difference. And that's so variable that will make any evaluation kind of shaky. That's the experience we have gained over the year. Again, if you have a good data, welcome to present to FDA. We are really open-minded on this issue. If anything doable, we do not need to proceed to CFD modeling, which is more expensive than doing those scale analysis with a smaller sample size, easier to manage. So that's a plus for everyone.
I think of the folks who discussed the methacholine, it is an agonist-antagonist approach to looking at pharmacology. It's the basis for all pharmacology, right? So in that context, what is the delivery of the methacholine? We had discussed some things of delivering by nebulizer versus some other way to deliver the methacholine challenge initially, and then followed by the treatment with whatever you're trying to treat with in a form of an MDI. And are you appropriately adjusting the doses? Are you on the same part of the S curve for a response? So that becomes a big question. And if you're not doing it right, then you're off, and then you'll miss any antagonistic effect altogether.
I think Bing has a question over here, or comment.
Yeah, Bing, go ahead.
She'll get you the microphone so online can hear too.
Thank you. Yeah, I just want to add a comment to the methacholine challenge. I think one of the challenge that with the methacholine challenge study is the missing data. You know, you can only give certain level of methacholine to challenge the patient, and beyond that, you know, you can't, right? It's a kind of a safety concern. So in that experience, in the experience that we had, there are lots of missing data where, the patient cannot get their PDE 20, PC 20. So we had to use some modeling simulation approach, which is very successfully done to facilitate getting that data missing part in there. We also, meanwhile, I think we also evaluate the bronchodilation study, and we found there is no dose response with the bronchodilation study. So a bronchoprovocation study was chosen at the time. Thank you.
Although I will say that at one point, either study design was in the guidance, I believe. I think it was only recently that the bronchodilation was removed.
The limitation study was that was removed from PSG. Then based on the internal sensitivity analysis as being mentioned, then the provocation challenge that was still, I believe, still in the PSC at the.
So we have a couple of questions from online about specific study types and how they can aid in BE assessment. So the first is, to what extent the PIV or PDI analysis can help in BE assessment? So that's talking about droplet size and velocity. Do we think that's an important component to measure?
Can you repeat the question?
Yeah, the question is, to what extent the PIV or PDI analysis can help in BE assessment? I think this is getting toward when Bryan was giving the summary of kind of what is more key study, what's supportive, what we can get from different studies.
Yeah, I can talk to that a bit. So I think figuring out droplet velocity and droplet evaporation rates is, for MDIs, is critically important to understand deposition in the mouth, throat, and then the lung beyond. I think there's a question that we need to address as to whether that's used as kind of pivotal information or important information for BE, or whether it's used as a model input and then the model predictions are used as that piece of evidence. And I mean, personally, based on the experience we've had on that, I'd lean towards the latter, where you measure these things and they're viewed as supportive information, but then they go into a model with lots of other different pieces of information. The model balances all of these things in kind of a physiologically meaningful way for the patients. And then it's that model output that's your BE evidence, or that part of the BE evidence. That's our perspective on that, I guess.
I think having done the same thing, I believe that these data should be considered what we call developmental data. Because if I use the word sportive, then comes another question. What are the limits for sportive? So as long as they are within certain range, if they are not within certain range, they are not going to give you any meaningful result in deposition and all that. But the question that I will have for PDI or PIV or PDA is the parameter that we use for velocity. There has been some question about the plume front versus the average plume velocity among the people who are doing it. And I'm not sure, but I believe that the industry recommendation has come from part of the work that has been done by the agency recommendation, part of the work that was done in St. Louis at one time for something else. And also what is for SMI, but the SMI is a aqueous solution and is a nebulizer. So one has to see which represents the most velocity. Is it because the plume in the MDI is changing as we go, right? It starts mixing. It's not like SMI. So therefore, that needs to be addressed. But yes, I would say developmental data. And then I leave it to the agency experts.
Go ahead. We want to hear from folks.
We talk about MDI, okay. So as we just discussed, if your Q and Q2 the same formulation, if the container closure of the system the same, the actuator the same, there's no reason to believe you'll get different velocity or droplet size distribution, prone geometry, spray pattern. So as a developer, you want to make sure those characteristics are similar to the reference product before you go ahead to do your spatial study, your PK study. We know if you have a different droplet size distribution, different velocity, then the chances are when you do the PK study, you are fair, you are not bioequivalent. So the important thing is that you need to make sure you design the product to be equivalent to the reference product. Then the testing and the measurement of the droplet size distribution velocity will be confirmatory. So as Dr. Zeng was saying, it's more development activity. Of course, if you have different formulation, then you needed to show that this formulation, different formulation, doesn't result into different droplet size distribution or velocity, because that will definitely result in difference in the PK profile.
I think Dr. Yang is, I absolutely agree with what you mentioned. If you have the same propellant, same size of canister, same valve of the same design and, meeting volume, same orifice, then it is the propellant that drives the speed of the plume, because the rest everything is very small there. And the propellant is going to exert the same. Most of the time, the supplier of the propellant is also the same. So therefore, I think theoretically, it should not matter, therefore development. But then the concept that Dr. Yang has just expressed, we should try to apply to the holistically to the overall alternative bioequivalence approach, that what are the things which could be so obvious and why are we going after certain things? So therefore we can make this alternative approach really workable and very appealing if we look holistically that we are using same everything, then why are we going into certain areas which could be like, if we are filing the hair, for example, right? And then there has to be a certain limit somewhere, but these are the parameters that go into your CFD analysis, and there has to be some relationship there. And in most cases, I don't think there's much difference between test and reference product. And we can't apply the same stat to this that we apply to, let's, for example, assay and single dose actuation content. Thank you.
Dr. Bäckman?
Just maybe a comment here. I guess from a kind of clinically functional perspective, it's still so that PK will be largely regulated by the quality and extent of dose reaching the lung. So I suppose that things like plume geometry and droplet velocity and so on is more kind of a tool for the developer to understand why didn't I get the lung dose out of, for example, a cast measurement or something like this. That is similar for the two products. So if you have that issue that you kind of with all respect for the variability in casts and the lack of standardization in these kind of realistic APSD method and mouth throat methods, I still think that that is a potential tool to assess the most clinically critical attribute of a product, whereas the rest of the data is more kind of supporting an understanding of why wasn't it equivalent or similar. So you can kind of go back and reformulate and try to get it to fit.
I have a slight different opinion. The velocity of spray that affect the coordination. We know from CFC to HFA transition, coordination becomes less of an issue because CFC, the spray come out much faster. So you need to coordinate it very well. So if you have a faster spray, it makes coordination more difficult. So that could affect the lung dose. Yeah.
Is that different? Is that shift actually?
You are measuring you're measuring the spray come out of there. So if it comes very fast, it makes the coordination much more difficult.
I thought our response time as humans were kind of slower than you can have in the spray velocity.
Yeah, coordination could be very difficult.
I want to make sure we hear from everyone. Did you want to say something? No.
Yes.
Some folks who haven't spoken.
Oh, yeah. Thank you. I think also depends on the correlation, the PBPK and the local regional deposition. If there is good correlation there, maybe the velocity and PDI may not be that important. But if the correlation is not good, those studies will be pivotal. Thank you.
Thank you. Okay, so we can shift. We have one more virtual question. Yes. Oh, I'm sorry. Would you like to speak more on that topic?
I guess just one point I wanted to, one thing I wanted to point out is that we talk about things being the same orifice diameter, same in vitro data, but two things are never the same. Like a test and a reference product are never going to be exactly identical. And I think by using all of the approaches together, modeling sensitivity analysis and stuff, you can figure out, you can answer that question of how different is tolerable, how different is required for bioequivalence. I think the assumption that things are exactly the same is perhaps slightly misplaced, in my opinion. That's all I wanted to say.
Thank you. So shifting gears a little bit, one question from online is asking, what about the human factors? We can ensure same or similar aerosol delivery performance and physicochemical properties of the drug aerosols between generic and RLD in the QT testing, but how do we ensure that these generic and RLD inhalers behave the same way in patient hands, which can bring some variety? Has anyone looked into it, and what research tool or tests can we adopt to ensure that human factors are also taken into consideration when developing generic inhalers?
So FDA does have some research in that space, and I don't know if industry has some interest in chatting about. And you had this said it very nicely, that MDIs, there are three main parts to it, right, that you want to look at. And the human interface is one of them in the context of the design of the MDI, looking at the angle. We had a whole bunch of talks about how that product is inserted or placed so that the patient can use it, and what guides do you have in that space to help them along. And test and reference products designed very similarly will help that to a great deal. And MDIs, there's not a lot of latitude for movement there, right?
Yeah, there is actually, particularly for suspension MDI. Of course, this is all part of development, part of a drug product characterization study. You need to show that, for example, suspension MDI, one thing you shake, after shaking, the time interval, the delay time, if you're longer shaking, longer delay time, of course, the delivered subsequent dose will be high, because if it's a good sediment, you have a sedimentary formulation. And if you have a creamy formulation, you shake, you wait for longer time, then subsequent dose will be low. So this kind of things, this misuse, conditions. If we do the drug product characterization study, we will show that our products are comparable also.
Yeah, I believe this human factor issue is more related to DPI than MDI, because there's pattern issue related to the design of DPI. But for MDI, I think the design is well established for many, many years. So it is common that the generic will use a similar design as the reference product. So human factors is typically not a big issue with MDI. I would say.
From my experience, companies that come in and meet with us for us to look at their MDI design, some of the main issues are, is the label legible? Because sometimes you've got an opaque holder, and then they might block the label of the insert, and that's a big problem. So those are things we easily remediable, but we do look at those.
Markham, let me give a comment, because I have been involved into this, both in discussion with the agency as well as trying to meet some of the requirements. It is a requirement now that you satisfy the human factor consideration. I'm not saying conduct the human factor study. And I will first address the first part that Markham has mentioned about the label, same label and everything. What happens is that you look at it's called threshold analysis, that you do your threshold analysis and submit to the agency. The threshold analysis identifies what are the differences in your device and the patient device, by including some of the people who may have used the inhaler or may be naive, and determine whether those differences are going to affect the drug delivery, which lead to safety and efficacy. Okay. Now, having gone through several iterations of different products, I think that's a very good method that the agency allows that. But when it comes to the human factor study, one thing is lacking in the guidance is the statistical method, certain criteria. Unless the agency specifies, for example, the D factor there, industry cannot do anything about this. They cannot determine how many, what's the sample size you are going to need. I mean, we faced this situation and then came pandemic. And when we calculated, it came to like 300 subjects or something, and finding 300 subjects under the initiation of panic when everything was closed was difficult. But I think agency was careful and very kind to consider the threshold analysis carefully, and we found that the study was not necessary. And it is applicable to MDI, DPI everywhere. But yesterday also we saw a big exception that usually we would think is not, like, for example, myelin device, which has to be inhaled vertically, somebody might think. But I'm sure that as it was mentioned, that the agency had made myelin do a number of studies to justify that. And so that was that era, but now it is established system. Bing, you have a mic in your hand.
Yeah, I just want to add some comment about the human factor study. Yes, Dr. Singh is right. Actually, the human factor studies serves as part of your submission to ensure the other aspect not being discussed in this workshop, which is the exchangeability and the device and the patient interface, that part, okay? And we have a guidance on that. And I think we also have a workshop, right? I took a quick glance of
There's a workshop on May 10th.
There is a workshop on November or December. Focus.
There's a training workshop on May 10th.
On that specific topic. So I would encourage the audience to attend that workshop. And for the myelins device, they did submit the human factor study, and their study shows that their device is exchangeable in patients' hands in their pre-submission phase study. So yesterday, I actually posted a question on what about, after the product was on the market, post-market data, and et cetera, trying to collect more information. Thank you.
Yeah, of note, the guidance that Dr. Singh's referring to is the draft guidance on comparative use in human factors studies for generic drug-device combination products. As we pointed out, that guidance is a draft guidance, and I believe it's on the list for upcoming revisions. So just to keep an eye out for that.
Yeah, but I appreciate we heard the two key input, right? One is in my translation, the margin for the error rate. So we don't have a clear number yet. The other is how to efficiently conduct study, either any other more efficient way to reduce the sample size. So we'll take note on that and potentially work something out.
I don't see any questions from the room right now, so I will ask one here. How can we translate or link PK data in healthy subjects to patients? Is there a way to rely more on the PK data in healthy subjects in lieu of the clinical endpoint study, such as by translating or linking PK data in healthy subjects to patients with, for example, asthma or COPD?
I could start if you want. I could just briefly comment. I think this is one area where there's a lot of areas, but one area where the FDA, as well as the other regulatory bodies, they got it right here. I think PK in healthy volunteers is the right way, because what we're trying to do is get the most sensitive model for assessing what's going into the body. So lung deposition, absorption, what's going into the systemic circulation. So I don't think you want the added variability of looking at different disease states. And I think that's reflected well in what the FDA has us doing and what the MHRA and EMA have us doing. I think it confuses the issue when we start talking about PK in disease states for the purpose of bioequivalence.
Okay, yeah. To me, I think the link from healthy to patient should rely on in vitro data. For example, looking at different flow rate for your cascade infection experiment. That could perhaps give you an idea as to how your product perform under different suction force that could be affected by the patient's status.
And I want to say that Ross mentioned in his summary of the session that he ran, there was a discussion there of doing PK studies in subjects who received the methacholine challenge. So you can have controlled bronchoconstriction with drug delivery. I don't know if that's feasible or not, but it was discussed as a hypothetical at that breakout session.
One thing about this question has been put on the FDA over the last 30 years or so, that you guys are doing studies in healthy volunteer, how you translate. But we have to look at what is bioequivalence basically. Basically, if you look at one of the chapters written by previous director Dale Conner, he points out that what is important is the release of the drug from the drug product at the site of action. Beyond that, it's all biology, which is so variable. So what we are controlling in the MDIs, DPIs, tablets, and capsule, what the agency assures that the release of the product to be available at site of action is the same. Now, for oral products, for example, if certain people have certain disease which built barriers in the GI tract, it's not the issue with the product. Because the drug is dissolved in the stomach, released in small intestine, and then if there's something happening later on. Similarly, with the asthma, COPD, or whatever other things are, if the drug is being delivered to the lung in the same dose, in the same sort of proportion, or with the C or P ratio, then once the drug is released out of the product, the body doesn't know whether it's generic or whether it's a brand. So it's going to have the same impact. So that's why it's the healthy volunteers thing.
Yeah, just yesterday when I heard the idea of combining methylcholine challenging with PK study, I thought it was a good idea. Then last night, after a few pints of beer, I thought it's a terrible idea. Why? The two reasons. Why? Because the methylcholine challenging study itself, we just discussed about how difficult it is to get the methylcholine challenging study right. Because we are using nebulizer to deliver the methylcholine to the patients. That delivery system is highly variable. It is not accurate at all. So that's number one. Number two, because the human airway is so sensitive to methylcholine challenging, so we may end up not detecting the difference between different test reference products. We may be detecting the difference between the methylcholine challenging method itself. So that's why I don't think probably it's not good.
But that was hypothetical. I guess someone needs to try it to prove you either wrong or right, whether they drink beer or not. Right?
Just one comment, Dr. Yang, I think you just underscored the reason why there is exclusion criteria about alcoholic beverages in making this, in conducting these bioequivalence studies.
I think he was sober when he was first discussing it, and then he reconsidered after the beers.
Dr. Liu, did you want to add something?
Yeah, I just want to add a little bit to what Dr. Singh said. The variability in the patient population actually can mask the difference of TNR products. Yeah, thank you.
Looks like one question here from the audience.
Not a question, just a comment. I think we do PK studies to detect differences, potential differences between test and reference product. And one should use the system that shows the highest sensitivity. So I always say that PK studies might be essentially a way to, or it's equivalent to an in vivo cascade impactor. And I want to use the most sensitive way of detecting differences. And I believe that's using healthy volunteers where you have the whole lung where it can deposit. If I have a patient population where, because of restrictions of the airways, the majority of the dose anyway just being deposited in the central region, then I potentially might not see differences between test and reference product. I think the most sensitive way of doing those in vivo studies would be in PK studies would be in healthy volunteers.
One thing I can add for the consolation of people who have concerns about not conducting studying in asthmatics, that the advances that OGD has made over the last 5-6 years by the inclusion of clinically relevant throats in the ABE approach at least, and then using the asthmatic profiles or the target population profiles. I believe that if we are using tiotropium, it will be the COPD patient profiles we will be using, where you use weak, medium, and strong profiles that represents mostly population. And furthermore, there are 3 sizes of the throats being used here. So that sort of, in my opinion, takes care of some of the concern, because the concern will never go away 100%. Some people will never believe whatever the agency says. But I think the stage where OGD is now with all this research effort, I think I have far more comfort than it was like 10 years ago. Thanks.
If I may just make a comment, if you go back to the kind of question, how to translate or link PK data in healthy volunteers to patient, and I kind of agree that one shouldn't do PK and with Günther and others, that PK in patients might not be the best way forward and it's not so sensitive. However, if one wants to explore the potential shift in PK and how it will shift for test and reference products going from healthy volunteer to patients, I mean, this is kind of done in clinical development using modeling. And I think there has been several presentations here from John de Backer and others on using modeling aspects to understand the shift in deposition pattern between healthy volunteers and patients on a, maybe not on an absolutely quantitative level, but it's seen on a qualitative level. What do the test and reference respond in the same way to disease-related changes in lung geometry and lung physiology? So I think there is definitely a room for various type of modeling to explore that area, which doesn't necessarily mean that you should run PK studies in patients per se.
Yeah, and I think on that, like, there are modeling technologies now that allow you to predict deposition or pharmacokinetics in patients quite rapidly. And the advantage of doing modeling and simulation in that is that if you've got a database of CT scans of patients, you can select ones to make sure you're covering the full heterogeneity of the disease. So you're not relying on random recruitment in a clinical study. So that means that if you were to run something like a virtual clinical trial or an in silico clinical trial, you could do it with much smaller sample numbers than you would have to in the clinic, and you'd get that kind of patient variability in the translation from the healthy volunteers to the patients. You could validate the models or establish credibility in the models from studies in healthy volunteers, but then run simulations to kind of do that translation to patients. And yeah, we can do that relatively easily now with the technology that we have.
Can I ask you a question following that? So yesterday I've been hearing the notion, using healthy volunteers. If I hear two schools of thought, at least I heard from 2 top-notch modelers. Question is whether if the product is behave equivalently in the healthy volunteer, would you also see them behave equivalently in the diseased population? I think that's the question I hear. The majority here, the opinion is not, but I want to get your.
No, let me, can I address that?
I'll talk first. So I think one thing that we discussed yesterday in the small group discussions was the interaction between the patient to patient or within patient variability and the product variability, and how you don't necessarily know which way that's going to go. There's some propagation of the variability in the dose to the lung and the exposure at the site of action that may exaggerate the difference between a test and a reference product. And without doing the work, we just don't know. And I think that's why my answer to your question is no, because I think we need to understand how these different levels of variability interact with each other. And there's no way we can say that without doing the work. So you can't make the assumption that they're the same.
Dr. Singh, do you have one final comment on this
Yes.
before we go to break?
Just one comment.
Sure.
This is a very important question. And we have to go back to what's the basic definition of bioequivalence is. I mean, there's a statute, there are regulations within which we operate. And again, it is the amount of drug released in certain conditions which are applicable in vivo and in vitro. And if those products are same, they are going to be bioequivalent in any population. Based on my experience, if you do in asthmatic, it's this question of sample size. If you do in COPD, sample size. In my opinion, we should stick with the healthy volunteer, not get into anything. People can suggest anything, come back. That is it going to be do this kind of modeling, do this kind of sophistication. In my opinion, that none of this is necessary. Why do agencies take a stand? That this is what we believe and that's what the world lives with, unless there is a gross mistake made by somebody, then the innovators are going to sue the agency. So far in the history of this, I have never seen it. So bioequivalence determined in healthy volunteers most applicable, unless we are working on some like skin disease or some psoriasis and others where you are applying directly to the diseased area. So therefore, when we start talking about going into very sophisticated modeling techniques, we again are coming back to only few people in the world who can do it. So let's keep it open door and see, I mean, in my opinion, there's no further work needed than going beyond the healthy volunteers. I welcome the panel discussion on that.
Thank you for all your thoughts in this session. We have now completed the first panel session, and most of the people will be back after the break. So if you have any more questions, feel free to bring them. We can continue the discussion. Thank you.
We didn't use it. No, and I still think that I don't fully agree that one should not pay attention to the disease state at all, because we are trying to waive a CEB study in patients. And so I do believe that one at least should show
Hi, everyone. The next panel discussion is going to start in about a minute. So please grab your coffees and get to the table.
Startup in down south. Not new to inhalation. Been in inhalation for 24 years.
Not many people left.
Yes. You can delete. I can delete some of the questions. Yeah, it's been interesting. I mean, everyone knows everyone. Optimization.
I sit next to you.
It's a great to meet you.
Hello. I'm doing well. How about you?
I'm missing William, though, aren't I?
Thanks. So we are on the same page.
You did that together.
Well, of course, but I mean, but we're talking about things that are trying to transport vastly through model
Just for chronic diseases, that's true. Acute, maybe not so much.
Yeah, I'm talking for chronic diseases.
Oh.
I'm thinking like the stuff we've been talking about at our conference, right? Like glutathione control for COPD.
Are you getting any more?
Yeah, we get some early development, early device development down in China. So we have a team of engineers in China.
Well, that
They are
changes
working
life a
on
little bit. Yeah.
prototype devices. So once they have done the functioning prototype devices, then we took the devices to UK, Europe, US, and to scale them.
We are full-time, yes.
So because the clearly, you can't manufacture devices fast, they can't meet so that's what we do. But we are very creative. There's the young people is really, oh my goodness, they have very, so they just.
That's what
You get a complexity of but there you get a complexity of you're trying to minimize. To minimize the cost of everything. Forming bigger reservoirs.
Start?
Right now.
All right, folks, so we're going to start our second panel session. You guys can have a seat in the room. For everybody that's joining us virtually, please feel free to submit your questions in the question box online, and then we can ask our panel at that time. So as a reminder, we just got a couple more seconds. Come up. Okay, so for our next panel session, what we want to really focus in on a bit more is on the dry powder inhalers. I think the first panel session focused some on the MDIs, but we want to specifically focus a little bit more about the complexities of the dry powder inhalers, some challenges associated with that, and then specific about how we're going to focus on the alternative approaches or what's the sort of challenges and research needs and gaps there. So I will start off with a more general question for the panel. Is in relation to the DPIs compared to MDIs, where do we see that sort of unique dosage form having potential differences or challenges in either the in vitro studies, the use and conditions, the conditions that may be used, or other aspects? So with relative to the MDIs, where do we see some of the unique challenges for DPIs? And if you don't mind, I might start, Abhishek, we can kick you off with more of an interesting perspective.
Yeah, so no, great question, and a number of us here have that experience, of course. DPIs are far more diverse, in my opinion, when compared with MDIs. The panel was discussing this earlier. Obviously, you have the canister actuator and the valve, but if you think of DPIs, there's so many more mechanisms of operation. There's so many more use steps, so many design possibilities. And for one, that makes the user interface very different. If you also think about it, there's also formulation, a diversity of formulation technologies you can use. You can have conventional DPIs, you can have engineered particles, you can have certain hybrid formulations. So I think that makes them uniquely placed when compared with MDIs in terms of complexity.
And then I'd like to ask another panelist potentially on what are some of, given that sort of complexity, are there specific sort of in vitro studies or conditions or ways in which we run these sort of studies that may be more important or more critical for a DPI formulation rather than MDI formulation? I throw it out there in terms of the realistic air particle size distribution. Is there a big difference there when the considerations for setting up that sort of experiment or conditions where we'd rely upon that? Diana, if you want to maybe start, I saw a smile down there, and then like I said, I'll open up to the rest of the panel too, to please feel free to jump in on that.
Yeah, so I mean, as we heard previously, the device is a lot more complicated for DPI, and on top of device characteristics, it also really depends on the patient inhalation profile and what they're doing. Arguably more than an MDI with a propellant. And so I think you have more challenges for realistic APSD as far as what is correlating to in vivo conditions. There may be more variability or more importance on inhalation profiles. So I think that's a space that can be explored unique to DPIs that may be a little more challenging than MDIs. It may be more clear, the in vivo correlation.
Sure. I think if there are any challenges, additional challenges, they are related to the device, that they are different devices. But what is important in the case of DPIs, that they are passive DPIs so far. And the testing has to be done on 3 flow rates. One are the recommended or what the innovator used, then half of that, and like 60-30 and 90 for Advair or Briolipta. And the other issue that sometimes comes is that we are doing cascade impactor, that the mouthpiece could be slightly different. Or the structure or the design could be slightly different, that warrants then use of a different kind of rubber adapter to that. And third comes the resistance, that it has a resistance. You have to do some additional testings. Once the drug leaves the device, then one additional step in the DPI is the pre-separator, which retains most of the lactose, like 99% of it. And once the drug is in the side of the cascade impactor, then there are no additional challenges, in my opinion, because it's then just washing it off and doing HPLC analysis. So it is that additional, we do 3 flow rates instead of 1. And that is the standard. That with regard to APSD testing, that's thank you.
I just want to chime in. In addition to what is already mentioned regarding challenges with DPI, of course, on top of the device formulation, actually the another challenge is manufacturing process. Because DPI powder blending, powder filling, this is a very complex process. Even for the same formulation, same API, same API particle size, if the manufacturing process is different, then you'll get a different performance and quality. That's number one. Number two, I know actually most DPI are susceptible to moisture ingress. So we want to make sure that, of course, the continuous closure system can protect the formulation from moisture ingress. So I talk about a patient, testing, so Dr. Singh was talking about, basically DPI different flow rate. Now, in addition to flow rate, because we know when we test APSD, we use a pump, very powerful pump, which reach peak flow rate immediately, instantaneously, which doesn't reflect the actual patient's inhalation profile. So it's even more relevant to use a realistic APSD profile to determine the performance of DPI products than actually the MDI products. Is what I just need to add.
I'd just like to add to that. I think there's a huge heterogeneity in the inhalation profiles that we see in both healthy volunteers and patients for DPIs as well. You've got different resistances, different device types as well, and that changes the shape of the profile. And we know it's not just about the peak flow rate or even the inhaled volume for DPIs of like blister devices like the Ellipta and the Discus. We've got some evidence that suggests that kind of initial phase where you're kicking the powder out of the device is really important. For something like a HandiHaler, where you're shuttling a capsule, then it's a different phase of the breathing profile that's critically important. So if you're trying to screen a range of breathing profiles to cover all of the important aspects, and there are different things that you need to consider just in terms of like the shape of the profile and the way that the profiles are applied to capture the right aspects for a particular DPI.
If I may, I'd also like to bring up the issue of a lot of the DPIs have multiple APIs, and that becomes an issue for a challenge for an analysis as well. And then what you said regarding the powder, it can settle over time. So storage, beginning of life, end of life differences as well.
And maybe one of the things to add to this is the fundamental aspect between the difference between MDIs and DPIs, the amount of payload. If you think about milligram quantities of payload in a primary packaging in a DPI, that poses its own challenges when compared with MDIs. And when you have a device metering version of a DPI, then obviously that has its own challenges as well. So here we are dealing with the complexity, and it keeps on evolving, and so you have to have techniques that are forgiving and can be standardized across the board.
So if you don't mind, I want to kind of get back into that idea of the inhalation profiles. I heard here specifically it's going to be an important sort of thing. What are the considerations, and what's the panel's opinion then on what are good inhalation profiles to start doing? I've heard there's going to be variability. Are we trying to mimic the patient conditions? What is the range that I think we want to look at for these alternative sort of studies, especially if it's going to start to help support something alternatively to the clinical endpoint study?
Yeah, I think the inhalation profile consideration comes into play at this moment based on the current guidelines or whatever the agency recommendations are, only if we are going into the ABE, alternative bioequivalence approach. And there it is the healthy volunteer as well as the target patient population, which in most cases is asthmatics, unless you are working on only LAMA product, that will be COPD. So there you have, let's say if you have the clinical study where for asthmatics we recommend 50 to 80% asthma, FE1 levels, predicted FE1 levels. Then you conduct a flow rate collection study from same kind of asthmatic population, right? You cover the target population and you get, let's say, 20, 30, whatever patients. And from there you, based on the lower and higher percentile, which can be 5%, 95%, or 10%, 90%, and then the median, that represents your asthmatic population then. Those are the inhalation profiles, which are different from, quite different from healthy volunteers. And then to address Dr. Yang's point, one is how you apply the inhalation profile and how much delay you give depends upon device to device, depend upon the product to product. So therefore, let's say if I'm using a capsule device and made one product, right? It doesn't mean that I'll be able to use the same profiles if some other drug product was put in the same device. So it is like Advair inhalation profiles are not related to Briolipta. And these are done with the placebos, not empty devices, these studies. Thank you. Go ahead, please.
Did you want to? No, I think I had more or less the same kind of suggestion, and that's the recommendation, at least to clients, to run a set of inhalation profiles in a relevant patient population and statistically select kind of the, for example, the 10th, 15th, and 19th percentile for studying the realistic APSD and the mouth throat deposition. I did have an additional comment maybe to the discussion of PMDI versus DPI in terms of testing. I think relevant cast models are excruciatingly important for MDI due to the ballistic aerosol properties and probably somewhat less sensitive when it comes to DPIs.
Maybe I can add to this is from an in vivo perspective, we typically think of asthma and COPD. So COPD not really flow rate limited, but if you think of a disease state in cystic fibrosis, highly flow rate dependent. And so you definitely have to look at your DPIs and their ability to, basically with the patient maneuver, ability to fluidize and deagglomerate both with a very low flow rate. Obviously, there's a volume factor that is there as well. So I think that it creates a complexity that you don't have in MDIs, that you have to deal with in DPIs to cover these indications and these broad patient interfaces.
I have a follow-up question to Dr. Tsang, also like a flow rate question. So you mentioned earlier that the manufacturing process can change the powder behavior. So in terms of agglomeration, deagglomeration, is there any learning, like how could the same powder but manufactured differently? How could what are the process parameter are you controlling? What should it be? Can that be reflected in the final product, like through in vitro testing? That's a follow-up question.
Is that a question for me?
Yeah.
Okay. That's a very complex question. Probably I'll take several lectures to address that simple question. And we know that during the manufacturing blending process, for example, there are many parameters that affect the blending, the homogeneity, and the energy put into. You don't want to blend the blend too much. Then you don't want to change the physical chemical property. You don't want to introduce a lot of high energy into the blend. So you really look at how to optimize the critical manufacturing process, critical process parameters, and understand how each critical process parameters, such as blending speed, and how that related to the final blend homogeneity and the final APSD profile, and also how each critical process parameter interact with each other. Then so you really need to use the design of experiment approach and really fully understand how each process parameter affect the finished product critical quality attributes. So I think typically you may have to make many blends at the lab scale and at a pilot scale and a commercial scale. So I'm sorry, I don't know whether I address your question, satisfactorily.
So how could you control the finished product? What in vitro quality or testing would you do to make sure that they are manufactured in the way you desire?
Yeah, that's a very good question, because now science has evolved so much that we really have very in-depth understanding of the dry powder inhaler formulation quality. So I mentioned it this morning that we define a quantity targeted product profile. That QTPP actually lists all the critical quantity attributes we believe are relevant to safety, efficacy, quality of the finished product. Then we link the manufacturing process parameter with those quantity attributes. For example, give an example, if a blending speed, and if your blend speed is too low, we know the homogeneity will be poor. If homogeneity will be poor, then your dose kind of uniformity will be poor. But if you blend too much, then you increase the interaction between lactose and the API, then APSD will be poor. So we understand, we need to understand the interaction between this process parameter and the quality attributes.
I think the very comprehensive
Yes,
response that Dr. Yang has given. The only thing is that the manufacturing process of the test product is going to be somewhat different than the reference product, because we don't know the exact lactose grade. If we know the supplier, that is also difficult to know. And what are the proportion of the fine and the coarse lactose we are using? It varies. What happens that it starts with a lot of science Dr. Yang has mentioned, but if you look into what happened in the kitchen, how our mothers became the best cooks, after a certain time becomes an art. So there are checks and balances here. For example, Dr. Yang mentioned about the blend speed, blend time, and the intensity, and that depends upon your lactose mixture and all that. But then all the tests that are generally applied to powders before even tableting come into play, blend uniformity and other tests. And then are all the chemistry tests given in the MDI, DPI guidance that are applied. But the key word he used is DOE. It's the design of experiment at the R&D level, which when you scale up might change slightly. Then you select, this is the formulation, then you go into stability and all that at a certain temperature and humidity, assure that throughout the life, that the assay, characterization factor, all that will remain the same.
So I'm going to jump in here to make sure we kind of keep back onto the DPIs and sort of this discussion. And before I turn over the audience questions, I know there's one thing that maybe it's kind of alluding to now is we've talked a bit about what's going into the capsule, the lactose fines, and the drug sort of components there. So where do we see in the alternative approach the need or the studies that may be needed of characterization of actually pre-actuation? How important are the pre-actuation properties? If so, which ones would be important to characterize and control? Or is this something that the post-actuation is of more importance, especially with relation now to the dry powder inhalers?
Darby, for the dry powder inhalers, I just want to point out that there are different kinds of dry powder inhalers, as was pointed out in the very beginning of this session. There are dry powder inhalers where the product mix is going into the capsule itself or to a single blister pack. But there are also dry powder inhalers where there are multiple blister packs emptying into a pre-actuation or an actuation reservoir that's mixed during product delivery. So that adds additional complication that the device itself has to be involved in the mixing, that the design of the device has to adequately mix the product in a similar way. So test and reference considerations then come into play there as well.
Yeah, let me take a stab at your question there, and it's a complex question, and it's very relevant to DPI, as we had a very good panel discussion earlier today on the pre-actuation assessment of DPIs versus MDIs. So clearly, hands down, I think it's more relevant to DPIs. Let's take the complexity of DPIs. The agency has approved two basic type of formulations there. One are the conventional lactose blends, the other are engineered powders. A majority of times they end up being spray dry. Now, high-density powders, free-flowing powders, low-density powders, not so free-flowing. So that creates a complexity. You also have excipients that behave very differently. So now you have this bulk interaction of your API, your excipients that are very different with the air as a media or as a solvent through the manufacturing process. That creates bulk powder movement. And bulk powder movement that transit can create a number of challenges. It can lead to triboelectric charging, it can lead to loss of fines, it can lead to enrichment, which is related to loss of fines. It can lead to improper flow, bulk material flow. And so while you're filling into your primary containment or your device, that in itself can be a root cause of a number of problems that are downstream. So I think the pre-actuation assessment, the better in-process controls that we can put would dramatically increase the quality of the products.
I wonder if the question was related to pre-actuation assessment of the finished product. Was it that, or is the development of the product?
So I think the question goes to is the product before it gets administered, but it's in the finished sort of drug product, what's the critical, so currently we ask for a number of critical quality attributes or tests, post-actuation, spray plume geometry and APSD. But
Abhi and Dr. Yang has addressed very much related to all the things that can contribute to. But once you have the finished product, you labeled it, whatever you did it, then comes the device issue, which the agency guidance, it lists a couple of attributes, like how many steps to open.
Right, I don't think we're talking necessarily about that. I'm asking about, as part of a alternative to the clinical endpoint study, how potentially is it important, or could it be a part of that sort of waiving of that clinical endpoint study to actually controlling and characterizing the powder that goes into the product, what's sort of the role of that sort of component?
If your device is same geometrically, the steps to actuator are the same, resistance is the same, your cascade impactor is the same, in my opinion, ABE does not call for anything else. It's like, it's more of a human factor issue than ABE issue, because what we are asking, for example, if there is syrup bottle available and say it has a child resistance lock on it, and are these different? It's the same. But we are talking about, in my opinion, none, but I welcome everybody else's comment.
Yeah, I think I'd probably agree with most of what you said there. I think if we can use some combination of techniques to identify what the most important product characterization attributes are, be that realistic APSD testing, dissolution of captured aerosol dose, microstructure and so on. If we can establish that those are the same between test and reference, and everything that happens pre-actuation, I don't think is relevant to an average bioequivalence approach. And I'd kind of apply that to PMDIs as well, talking about sort of suspended particles in cans and things like that might be asking the wrong question, in my opinion.
I just want to add something. If I'm to develop a capsule-based DPI, of course, the first thing we'd like to do is to analyze what is contained in the capsule, the lactose API, to make sure that I'm Q and Q2 equivalent to the reference product. And also would like to characterize what kind of lactose, or the API, the capsule, it's a HPM capsule or gelatin capsule, what water content in there. So I will do a full characterization from a development perspective. But the really relevant for BE is a Q1, Q2. And also I would like to understand, the particle size or particle shape of the lactose or excipient, if I can use MDRS, and to fully characterize so that I know what target API and excipient particle size or shape I'm going to achieve. So that's for blister strip similar. First thing I would like to do is analyze the Q1, Q2, make sure Q1, Q2 is equivalent. And also we can use, I can analyze the particle size of the bulk formulation and also surface area, DV, etc. Is to support me to develop, identify, to make sure my formulation is as close as possible to the reference product. So they may not be part of the BE, but the Q1, Q2, and the Q3 that can be part of BE.
I think the industry have maybe spoken and said that we don't think that pre-actuation is important. I wonder if we get some perspective from the agency on that as well.
I'll just offer a provocation.
Okay.
And that is, if you consider a dual API system, for example, the one we've talked about a lot today and yesterday, the fluticasone somatropol system, where the two drugs are known to be very cohesive with each other. If you have it mixed differently, then the same actuator will in fact not take the two different pre-formulations and give the same answer. I'm giving a particular example, but it's an important example. And we know what the cohesive interaction is there. We already know that they preferentially stay together. So I think it's not, I don't know if it's cut and dried.
I think that is taken into consideration when we develop a DPI. This cohesive adhesive properties are to some extent taken into consideration. But one also has to take into consideration what's the cost of those. And the main thing is that at 3 different flow rates, when we are testing, your output is per the label of the reference product. Your particle size is the same, right? And then whether you have a different kind of a cohesivity or adhesivity and it just remains the same over the 24-month stability period, then this becomes a moot point. Then because all that is taken, that's why I asked this question, is it related to finished product or are we talking about manufacturing? I think question is related to finished product. Thank you.
Yeah, I would kind of assume that if there are any major changes in API interactions in the bulk material, you will actually see that in the stage deposition of the individual drug components. There was one other comment, basically, we haven't discussed, and that is probably that one needs to understand the release of the bolus drug aerosol within the inhalation flow. If that varies between products, even if the essential APSD, etc., is very similar, you might get some differences in deposition pattern.
Can you elaborate your question that what could be different release pattern from the DPI? When you test them same at 3 flow rates, why do we need to worry any more than that?
I was more considering if different DPIs or test reference products have a different release rate of the drug, for example, from the capsule or from blisters, that they come immediately in the inhalation flow in a very short, small bolus volume, or if it's more expanded bolus volume, or if it has a delay volume before you inhale it. then it means that the aerosol particle actually hits the obstacle, so to speak, in the airways at a different flow rate as you change the flow rate during your inhalation.
I think that's where the differentiation between the finished product and Dr. Singh, as you said, the differentiation between finished product and process comes into play. If you've done a good development, which is quality by design, designing an equivalence by design, then you would have determined your control space, you would have determined your design space, and that would address the topics of the nature that we're discussing here. Yeah. Of course, this is expected. You will have batch-to-batch variability in the API, excipients, and a number of other parameters. But that's exactly what you evaluate in your DOEs.
Yeah, I also want to make comment that following both of the comment from here, they said, I think what I hear, if you say if the in vitro particle size, morphology, or the surface charge, all the parameters are the same, you may speculate the flow with different flow rate. There's ABE. So the question is that have we collected all the parameters that will be biopredictive? Even there could be potentially hidden parameter, by morphology, particle look the same, but the density could be different. The separation behavior from the aerosol could be different. No, I'm not saying I'm a very bureaucratic agency employee, but so we have to figure those out. I think a modeling at this point do offer opportunity for us to evaluate whether those in vitro testings are bio-relevant or not. Yeah. If so, by how much? And we have to say, yesterday, Elon Musk launched a rocket, right? Exploded. I think he calculated everything should be correct, but still exploded. There are some hidden factors we need to figure out, before we really say the in vitro testing will be perfect, will be sufficient. That's some comment, more like a.
Yeah, I just want to add to that, and with regards to differences that you can detect, I think there's some evidence that even if you have the same flow rate, you may get different measurements of, say, dose delivered, depending on the apparatus that you use, depending on what USP apparatus you use, what other criteria you use in the context where you're doing the study even. So there is some variation with these DPIs. They're a little bit tricky in my consideration.
I just one comment regarding that. Number one, there are several properties of the API and the lactose which are characterized, and the DOE designs are in place to determine which are the most important that affect the powder and the delivery and all that, right? Like surface charge, surface roughness, things like that. There are about 10 of them. Liang, we also have some modeling in place. When we have enough data, you try some modeling exercise to determine or to validate our assumptions. Beyond that, I think it's a never-ending list.
Yeah. Could I make a comment on that? Yeah, I think it's a very valid question. I've worked on inhalation for over 30 years. To me, the most sensitive parameter of critical quantity attributes from a DPI formulation is actually, or even MDI, is APSD profile. APSD, measurement by NGI. That, the APSD profile is determined by a lot of parameters. Anything, if there's any change to the formulation, to the process, to the container closure system, even to the testing conditions, that is reflected by APSD profile. And luckily, APSD profile is the most relevant profile, to the lung deposition. If you can get APSD right, That means all the other parameters, no matter how they will affect the performance, their combined effect will not change the safety efficacy of the finished product. So I give you an example, when I was working for a company before, we had a QC testing lab in Ireland. And there was one analyst, always got the OS results. And we did a lot of investigation, training. We found out that analysts generate static charge on the lab code. So that proves that the APSD is the most sensitive parameter and also most relevant parameter. So that put me at ease that no matter how much variables that could be involved in determining the APSD profile, as long as we can get APSD right.
And also,
Can I
to
open up
address
the
Markham,
questions to
just one minor comment, please. Markham raised a very important issue here, is that despite doing all that, then what happens if some different conditions come, right? Let's not forget that there are CMC guidance for MDI DPI has a long list of rigorous characterization studies that the test product has to go through, and most sponsors also run the reference product through that. And that is temperature, soil cycling, shipping, moisture, this and that. So it goes to a lot of rigorous, not just ABE testing, the test products. And I agree 100% with Dr. Yang is that if through all that, you have the same emitted dose and APSD, and then there is no concern beyond that.
So I would like to see if the audience has some specific questions for our panel to start off.
All right, thank you all for participating today. I want to ask our non-FDA people a hard question. So when I think about the dry powder inhalers, the most worrisome thing about thinking about our new alternative approaches is, as we all know, lots of public evidence that the PK studies, right? PK is an important part of an alternative approach. PK studies have a lot of batch-to-batch variability. All right, so I'd like you to, all of you non-FDA people, to comment on how you would address that during development. Do you think that batch-to-batch variability is predicted by the realistic APSD studies? If not, then what do you think the cause is? Because it seems like that cause, I think people said someone said surface chemistry doesn't matter once they measure the particle size. So if I feel like we have to be able to explain the batch-to-batch variability with something measured to really say we have a strong, in vitro alternative approach. If you say, well, now I know why there's PK batch variability, and then I can design the product. So I really welcome sort of the comments on what you think the origins are and how you would address that during your product development processes.
Yeah, let me take that, Robert. A great question, actually. So the challenge that you mentioned is, in my opinion, is an opportunity. It tells you how sensitive the PK can be as a methodology to test even the minute differences. One of the reasons we actually sometimes design that on purpose, in fact, all the times we would design that on purpose to vary things and study them in a pilot PK, let's say. Now, it's obvious that you see those, excuse me, the batch-to-batch differences or the API differences or the process differences. And that's by design, and you have to be able to control that through a holistic assessment of your control and a design space, as I mentioned earlier. So it goes to good development. It goes to understanding the fundamentals of the material you're putting in, the process you're developing, and how you're controlling it through the entirety of your supply chain. And obviously the control and execution of the study comes into play as well. But I think it's an opportunity. It's not really a challenge. And that tells you that there's all the more of a reason to make PKBE as the primary goalpost for us.
I'll give you a hypothesis. The hypothesis you could state is that the mean physicochemical data is not indicative batch to batch, and that perhaps what's missing, especially in the multiple API systems, is an understanding of the distribution of physicochemical properties across the particles, which is hidden in the mean data. And so that in fact, if one had that information, one wouldn't see variability.
Just want to add to that, the evaluation I've personally done, and we've done in terms of understanding batch-to-batch variability of both reference and test products, normally boils down to measurable differences in things like fine particle dose, lung dose, and so on, that could come with age and variations in manufacturing and so on. The paper by Burmeister gets that show the batch-to-batch variability, I believe, on Advair, and we did an analysis for that for RDD a couple of years ago and found that the difference could, one of the batches were 1 year older than the other, and the differences observed in PK could be explained by a 10% shift in fine particle fraction, which I think is not unbelievable in over a 1 year storage period. So.
Just one very brief comment. I think we shouldn't forget another in vitro system, that's the dissolution. One should also look at the dissolution rates, not only at the particle size distribution.
That's true, but in my experience, the dissolution rate seems to be more stable than the particle size distribution over time, for example, and between. But that is obviously depending on manufacturers' controls because as you mentioned, the dissolution rate would be extremely sensitive to changes, for example, perhaps in our API supply and so on. So one would assume that these checks and balances are in place when.
But in our study that we did, I was very surprised that lactose fines can modulate dissolution rates significantly.
Absolutely, and that's kind of shown in the differences in specific dissolution rate for the 3 Advair strengths, where that's quite clearly shown that the difference in kind of Cmax over AC is explained by differences in dissolution rate. That is, as I think Will and colleagues demonstrated a few years ago, that could be related to whether an API or a fluticasone particle was close to a fluticasone particle or close to a micronized lactose. It has a large impact on the specific surface area as it deposits.
And I think within the context of an alternative BE approach, if we do see large batch-to-batch variability in PK in the reference product, if we do a full characterization with some, I guess, both bottom-up CFD, PPPK, but also pop PK modeling, and we can explain those differences. And I think that just lends some power to the alternative approach and could actually help us. We spoke about doing these kind of assessments in products that are designed to be the same versus products that are designed to be different. If there is some batch-to-batch variability, then it gives us an opportunity to show the sensitivity of the method as well. If those relationships do indeed exist, which I guess we need a bit more evidence to look into.
Do you have a real case?
So, can I add in just there's a bit of experience here, but it does go back to what you're saying, Will. Is the first step in any of these programs is characterization of the RLD, and that will include getting some pilot PK data on the RLD, and then using that to validate what in vitro measure is going to be important. But part of that is getting a number of batches in and characterizing and keeping it on shelf life and repeating that process. So you're building an understanding, and I think that leads to the challenge of what the PK bioequivalence approach is, which is that one time point, one batch against one batch, and you're trying to move, you've got these moving targets, the 2 trains, if you like, on 2 tracks, and you're trying to balance that to get to that point. Now that is one way of passing a PK study, but with all of that knowledge base you've got behind it, which realistic APSD may help be a bit more sensitive to understand where those changes are coming from, what the parameters are. But I think that can help establish a sort of boundary conditions where that PK study is then appropriate. And maybe that's sort of that totality of that experience that needs to be looked at as much as the single one study point. But it does come from understanding what you're trying to match.
So you're saying be smart in picking your reference batches?
No, it's being realistic that you've not got an outlier in the batch that you're picking for that study.
If you have an in vitro study that you see, like I said, I think the importance of characterizing not just the PK variable, but also the sort of more advanced characterization of those batches, that's the thing that sort of shows you, oh, this PK variability, the cause of that is I have some evidence, oh, it's this part of the particle size distribution is the difference. And then you can think about more rationally deciding what the right, because it is it's a challenging regulatory issue too when the reference product has the variability of coming up with a reasonable and fair standard that also really gets to the right target about what the patients, are expecting. But I think if you have that understanding of this is the aspect of it, then your study comparison can say, well, look, I'm comparing this product to here, and they match this, and then they match that. And because there's, but if there's variability in somewhere else, then that's understandable, predictable, and then it's not, if it's predictable, it's not so worrisome.
I think, Rob, what you mentioned, at least that is the practice in my department. Let's say, initially we do some exploratory pilot studies and to see where the formulations lie. And then if we do DOE, I mean, there are only so many studies one can conduct. Then when we see difference, we go back and look at various parameters used. We look at particle size, we look at the moisture content of lactose and other stuff. So try to pinpoint what was the cause. And if we find a root cause, then you try to replicate again, whether it is going to reproduce or not. So, but sometime when it happens with the reference products, that's where the real challenges can come. And that what they have done in their manufacturing of that batch, we don't know if they, but with the test product, that's our standard practice, that if there's any change that you see in vitro, even before you go to PK, and then sometime what you find in vitro is the same and somehow PK is different, then you have a number of other avenues to look at, whether it was a dosing error, whether it was something else, or moisture regress. So it's a challenge, but number of avenues are explored to answer those questions. Thank you.
Is there additional questions from the audience? I think
online, no questions.
Yes, there's one. I think it's very specific. The question we received from online. I'll read it out and make sure it makes sense. It's discussion about the rapid prototyping and conducting realistic in vitro studies, and a question asking about developing of low-bar drug deposition setup as supportive in vitro evidence for ABE. And specifically, I think they're asking a little bit more here about the use of that as a study or test in terms of the low bar. I didn't know if that was, we tried to get a bit more clarification of the question.
Can you explain as to what the question is? Yeah, can you?
I threw that question out and I've tried to interpret the question. That was from online. We've tried to ask for a bit more. So I tried to throw it out to the panel to see if the panel could interpret the question. I can mention it once more if need be. I did want to say that. You mentioned about the use of rapid prototyping and conducting realistic in vitro studies. Have you come across developing low-bar drug deposition setup as a supportive in vitro evidence for ABE? If not, what would be the perspective on developing one?
Darby, I think I understand the question.
Oh yeah,
I think what they're saying is that they've got like an in vitro whole lung setup rather than just a mouth throat model. So, but apparently they're measuring deposition per each lung lobe. So you can do with that as you will, but I think that's what the question is.
Is that with radio labeling or?
More like a standard mouth throat model, so you wash it down afterwards. I think if the person that asked the question has one of these, I'd quite like to have a play with it or because I think that I had a discussion with possibly Ross yesterday about this, where I think there are some challenges in doing CFD, for example, in CT scans of lungs and getting that information quickly. If there was an in vitro study that we could do, well, yeah, rapid fast screening of different things in the laboratory, then that could be quite advantageous. And I think if there was a system to do that robustly, then it would definitely form part of an alternative approach, in my opinion.
So there's still, what I'm hearing from you, there's still a lot of gaps in that sort of development required, if it is, or proof and validation
Yeah, I mean,
types of things?
there was a paper VCU published quite a few years ago where they essentially had a cast of the first few generations of a lung. It was idealized branching tubes inside like I think it was a perspex or a glass cylinder, and they attached a pump to it and pulled through, washed it down afterwards, and they were able to get an idea of what deposited where. It's a nice idea, and actually we're seeing it more and more with nasal studies where we're looking at nasal castes that come apart into different regions of the nose, and they've been incredibly useful. So I think, yeah, that technology is something to consider, but I'm not sure whether...
all I can say that we did develop a system, and we have a patent on it, and I think patent is available, people can read about it. I can't go beyond this. Okay.
Is it in your name?
No. Whether it's in the company's name, let's say, the people who spend a lot of time developing it, but it's a huge amount of task. Let's put it this way: if you go to generation 5 or 6, and then you have different kind of lobes, right? It all depends that how close is your system to in vivo. Then it's always question. But we ran, let's say, a couple of drugs, and we found that it was pretty close to scintigraphy data. Thanks.
Then I'll kind of shift then actually to in vitro and vivo correlations and a bit more of that about where we see that's going to be potentially desirable, important, or we need to potentially add more growth or research. So when considering and developing an in vitro or a model integrated approach that's an alternative to the clinical endpoint study, what are some desired correlations between those alternative studies and the in vivo performance of the drug? And Per, maybe I can start with you as some thoughts around that in terms of the IVIVC needs and desires.
I think one attractive correlation to dissolution is actually the Cmax over AUC ratio, because that in itself has kind of an inbuilt reference. So the specific peak sharpness should be quite well related to the release rate profile in the lung. I know it might be affected, of course, by deposition differences as well, but the AUC is not very dependent on the dissolution profile, but the Cmax is extremely dependent. So that could be one element of correlation between an in vitro property and a PK measurable property that one might want to employ. Otherwise, I think we heard yesterday or today a discussion on of course, deposition pattern versus fractional AUC, which I think was a quite very interesting concept and idea to be done. I think I'll leave it at that and see.
Is there additional comments from the panel? I think Diana, sorry, I'll let her go give a quick shot.
Sorry?
Diana, about the end.
I can just make a quick point. So I think anytime you have in vivo data that you can use to kind of justify the conditions that you're using for your in vitro data or to validate your model, all of that is really important in an alternative approach, because really we're replacing the clinical endpoint study. So we're hoping to see real patient variability that we would expect somehow reflected in the in vitro studies used. For example, I know we were talking about different patient ranges for inhalation profiles. Another way we've seen this, which is kind of somewhat convincing, the argument is to look at what we've seen in scintigraphy for total lung dose and comparing that with, we have these inhalation profiles or these mouth-throat models, what actually is correlating to in vivo data that we would expect from the intended patient population. So I think that is really valuable to kind of place importance on maybe in that space.
I think you're absolutely right. What I was going to comment is that the best opportunity to develop a in vitro correlation is between the APSD and the PK profiles. Beyond that, I don't think there's any opportunity. But anything representative of the patient population, as you mentioned, then I think one has to look at, for the standard BE, it's only ISM, but I think when we want to develop ABE, we do look at lung dose and even the peripheral and central into the different flow rates with the different patient profiles for the target population. That's all you can do about this. And I'm not saying that there will always be a relationship. There may be a relationship always with the total lung dose. If that doesn't happen, then there is something wrong with it. But for certain drug products, whether C over P has a direct relationship, we don't know. But then I can also mention that for a given drug in a different formulation, which there are two products of beclomethasone out there, which are so-called fine particles. One is QVAR, the other is Clenil. And Clenil gives a particle size of about 3 microns, and QVAR is about 1 micron. And there's a very good correlation between, regional distribution, and because of the formulation differences and some device differences. And that is very well reflected in the PK. But if you look at, there are more than 5 studies in the literature comparing the effectiveness of these two products is the same. So the opportunities remains, and there's much more empty deposition in case of Clenil, and it's reflected very well into the PK data. So that's where I think it comes. Yeah. Thanks.
Hi. So it was pointed out by several folks in the audience over the last 2 days that FDA has the privilege and the responsibility of accumulating the data from various clinical studies. And so pulling those data and using those as the basis for modeling for IVC, looking at those data and interpreting them, doing cluster analyses, like I saw some really nice cluster analyses done with regards to what are the patient pools, what explains the variability, and then bringing that back into, okay, well, can we tweak the in vitro model to adjust for those variabilities, and are they clinically relevant in the context of of product development? So product development can be split into, and for us, we're very interested in BE. So we're looking at product development towards the BE range, but there are other opportunities for that data as well with regards to new drug development, et cetera. But back to the BE thing, we're looking at best models for doing those. And we have some fantastic folks here that are combing through those data coming in, and we constantly get new data to look at, right, Liang? And so each time we get that, we can add to that model. The more that goes into it, the more we understand some of the complexities and maybe and also accumulating in vitro data at the same time to try to correlate the two. It's a tough task. I know that it is, and it's good to have bright minds in the group like all of you to think along this with us and to contribute to the field. This is a growing opportunity for society. So this is great.
I was going to say, so I mean, we saw today in the in vitro session some really beautiful video rate data out of University of Sydney that is really exciting work and ways to think about connecting that. And wonderful talk from FDA on MDRS and the ways to connect that. And there's a lot of other new methods that are out there which allow us to take methods that are currently bulk, but we could now measure those properties at the particle by particle level. And it doesn't matter, right, to the in vivo connection. And I think, we won't know till we look and try.
Okay. I think this is pretty close to the end of our session here. I want to thank everyone on the panel for their contribution to the day, and I think I will hand over to our closing remarks for rest. So thank you, everybody.
So I look forward to the follow-up discussions.
think it's key. There's not much in vivo data other than PK.
Oh, yeah.
Image, but nobody want to invest on the imaging.
But they want
So you've got a lot of in vitro and a lot of PK, but,
yeah, but no,
not much else.
you're not going to get and also it may not correlate. It will correlate with this everything.
So here too, we come to the point of ending this workshop. 2 days program, 21 presentations, extensive in-depth discussions. How exciting. Can I have the second slide, please? Yeah, first, I'd like to take this opportunity to thank our CRCG members and our FDA colleagues, especially the organizing committees. It is your dedication and your hard work. Many of them are behind scenes that put this workshop successful. Thank you. And I also want to thank all of our speakers. You know, it is your very insightful, informative presentations and your knowledge and your experience that shared in these presentations provided a foundation for the future considerations of the alternative approach in this very complex product. Thank you. And a big thank you goes to all of our panel members. Your point of view, your insights, your expertise helped demystifying some of the complex concepts and also deepen our understanding in this rather challenging area. I've been so much enjoyed in this discussion. Thank you. Lastly, I want to thank all of our audience and attendees, whether you attended through online or you make the effort to come here in person. It is your active engagement, your participation, your thought-provoking questions, and your valuable feedback that brings to a very lively discussion on this topic. Thank you. Okay, yesterday, can I have the next slide, please? Okay, yesterday morning, we came to this room with a theme, alternative approach to comparative clinical endpoint study and a PD/BE study for generic early inhaled drug product. And some question marks, when, what, and how. So today, at the end of this program, where are we? So last night, I was trying to sort out some of the gainings, observations that I gained from this workshop into two buckets, okay? One is the guidance development bucket, and the other one is the guidance implementation bucket. I'm trying to put the questions, the challenges, and also the possible actions into these two buckets, okay? In the guidance development bucket, I listed 3 questions in the descending order of clarity. First question, are we going to go with this alternative approach? I think the answer is clear, yes. Second, do we gain a better understanding of the advantage and limitations of this series of in vitro, in vivo, and in silicon approaches discussed in this workshop? I also put a yes here, especially for those very critical tools that brought up for the discussion and explored in depth. Through this workshop, that could turn into a yes. And the third question is, with all of these gainings, with a better understanding of these tools, are we able to identify the critical tools? I put a critical and underscore in critical, okay. The critical tools as a holistic alternative approach for clinical endpoint study. I would say, in my mind, it becomes more clearer through this workshop. So the action in this bucket would be take the knowledge we gained from this workshop and develop, number one, more PSGs for this complex drug product. And the second, but more important, is to be more specific, develop the PSG in a more specific and defined way. Okay. So in the second bucket, I also want to share some of the thinkings that I put together. It is important to know that the in vitro method may not be always easier to be implemented than the in vivo method, especially at its beginning phase of implementation. Our BCS Weaver experiences actually tells us this. And also, I think many of you know the first generic memadizole nasal spray experience, right? We approved this product in 2018. This is the first product which we considered to use the in vitro method as an alternative for a clinical endpoint study for this complex nasal suspension drug product. And it took 8 years for us to approve this product. Of course, we don't want to see it in the future. So in other words, translating the scientific principles from the lab to the drug development, then to the product approval has been always a challenge that we need to know. However, the in-depth discussion in this particular workshop into the very practical issues has helped to remove the hurdles and the roadblocks for the future implementation. I can give an example. I've been travel back and forth in these 2 rooms and getting a sense of the discussion. In the dissolution session discussion, we discussed sink condition, method validation, we discussed the sampling, we discussed surfactant effect in the dissolution, discriminatory versus biorelevant. All of these practical points could turn into a deficiency if you don't put it in consideration into your drug development. And also, I think the collaboration between FDA guidance development team, I see, our big team, guidance development team is here, and the application review team, our office director, Dr. Pasaroy, he was here yesterday and he's here with us today, and also industry. The 3 parties collaboration is critical to bring the product to approval. Next slide, please. At the beginning of the workshop, I think Rob set up the tone for this workshop. He used 2 words, honestly and clearly. I like it. I went to him and I said, I like it. I would actually add a few more words into it. Openness, transparency, clarity. Okay. I heard honest and constructive feedback. I also heard instrumental suggestions. I heard innovative and stimulating ideas. I heard emerging technologies. I especially enjoyed the small group discussion where we drill down to the very deep and getting to the core of the solution. So I could still call it a transition time before we approve the first inhalation product using the alternative approach. But I also want to quote one of our speakers saying, "The future is bright." I firmly believe that the knowledge, experience, and insight exchanged in this workshop through the comprehensive and in-depth discussion will lead to a leap to the alternative approach for this generic inhalation drug product. And with that, I conclude this workshop. Once again, thank you for being part of this excellent experience. Thank you.